Hello and welcome to BIOS Builders. We are thrilled to welcome Kevin Parker, founder and CEO of Cartography Biosciences, to the show today. Thank you once again, Kevin, for joining us.
Yeah, thank you for having me. Glad to be here.
Fantastic to have you, Kevin. It's great to sit down and chat. Would love to kick things off. Could you just briefly share a personal introduction on yourself for us?
Yeah, so right before cartography, I was doing my PhD at Stanford and working in kind of the interface between genomics technology development on the molecular side and the computational analysis of that data. I actually didn't think I was going to be doing biology when I kind of go back even farther. Growing up as a kid, I hated biology. To me, it was something in high school, like you memorized facts in a textbook, didn't get it, didn't like, why am I memorizing all the stuff that I could just look on the internet? Um, but as I came to appreciate biology as a field where you're asking and answering questions, it really started to emerge as something that I enjoyed a lot. And so in undergrad was working in stem cell biology, developmental biology, trying to understand how cells make decisions. And as I started to think about what was going to come next, I wanted to do that, quote unquote, training as a scientist that you get as a graduate student doing your PhD. So I came to Stanford, was lucky to join Howard Chang's lab there and work with Howard and Ansu Satpathy and a few others. Initially, as I mentioned, on the technology development side. So how do we build new tools to measure individual cells, perturb them, see what happens? Um, I think that the academic kind of intellectual curiosity of that was stimulating and fun, but for me, it lacked a connection to patients and translational work. And so I started to spend some more time looking at immunology, immunotherapies, CAR T cell biology. And one of those projects was looking at CD19 directed CAR T cell therapy. So B cell malignancies and redirecting T cells towards cells that express the protein CD19. And that project, we were trying to understand off tumor toxicities. That led to understanding more about target biology and the use of single cell genomics, which led into some of this work around photography, around trying to find new targets that are more specific and more precise. So there was some academic through line, but I've also sort of bounced around between a few different areas over time as well.
Amazing to hear the introduction on yourself, Kevin. And as we're bringing up the core underlying technology that you so amazingly pioneered out of Stanford, I'd just love to start here just before we dive deep. Could you just provide a brief introduction to cartography for us?
Yeah, so choreography is focused on building a therapeutic pipeline around more precise targets. We're focused on antibody-based therapies, in particular asking the question, how do we make an antibody go precisely to the tumor and have its effect just on the tumor, as opposed to other cells in the body that you're trying to avoid? So about half of the company on the R&D side is focused on our platform. We have two platforms, our Atlas platform and our Summit platform. But both of those are really built around how we understand specificity to those cells that are driving a patient's cancer. So we've mapped out every cell in the healthy body, as well as every antigen that those cells express. So we have that reference. And then in addition, we build in these tumor atlases to get these cohorts of patients, same thing, single cell profiling of those samples, and have this unique database that then lets us find more specific targets to those cells that are specifically driving a patient's cancer and build a pipeline out from that. And that's what the other half of the R&D team is focused on, building that pipeline. So we're building antibody-based therapies, thinking about T-cell engagers as our lead program, but other modality platforms as well, in principle, ADCs, for instance. So our first program targets antigen and colorectal cancer. We're really excited about that, but have a lot of other things behind that as well in the works.
A lot of exciting things that we're looking forward to see along the growth and the exciting journey that cartography is continuing to have in some more recent news that I'm excited to dive into later in this
Yeah.
episode. But beyond that, I really, as we're at the Genesis story and you started to alluding to, I'd really love to just go down here to the origin story here, Kevin, just from your own perspective. And so, as you mentioned, right, in 2020, you helped co-found Cartography with a vision to enhance precision therapeutics for cancer via comprehensive antigen profiling to build safer and more effective therapeutics. Really just taking a step back, as you alluded a bit to your work at Stanford, could you tell us more about the actual genesis of cartography? I mean, what was really the spark that started it all?
Yeah, I think that there were a few things that came together to really kind of motivate cartography. The first, as I mentioned before, was this work understanding off-tumor toxicities of existing therapies, and in particular, asking why CD19 directed therapies had more neurotoxicity than did other B cell targeting therapies. So just to kind of unpack that for a minute, um, there were two papers in the New England Journal of Medicine that came out, I believe, kind of end of 2017 or so, showing really amazing efficacy of CAR T cell therapies targeting CD19 for B cell cancers. And what was interesting kind of intellectually was that these trials also reported a pretty high rate of neurological side effects. So patients had blood-brain barrier disruption, some bleeding in the brain, in some cases pretty severe, and in a couple cases fatal. And we were trying to ask the question why it was that CD19 directed therapies, so B cell targeted therapies, would have problems in the brain. That was kind of an unobvious linkage at that point in time. And in that work, we specifically looked at single cell sequencing data of the human brain. So measuring individual cells in the brain one by one, you have your neurons, you have your blood cells, you have your microglia, you have your blood vessels, everything in between, to ask whether we could find any cells that had any signal around CD19. And we actually found some cells that line the blood vessels of the brain that seemed to have some expression of CD19. Some other cells that looked like microglia potentially also had some expression. But it basically was this very rare cell population, kind of sub 1% of the cells in the brain that had pretty clear signal. So they were expressing CD19, but because the population was so rare, you missed it by other sequencing methods. And this made us appreciate, oh, there's actually a pretty big need here to understand these targeted therapies and the expression of each of those targets at the individual cellular level of every cell in the body. Because it's one thing to know you're targeting a tumor, but you really need to know everything else that you're hopefully not targeting. And so that was an academic project that got published, I think around 2020 in Cell, that made us realize, okay, first off, we have a lot of programs in development. They target a set of targets. How do we predict off-tumor, on-target toxicities more robustly, more quickly, more accurately, without having to go into a patient? So how could we have predicted if there would be any signal in the brain around CD19 expression? And turns out that there's actually a pretty big need there to build up better tools to kind of predict this toxicology around target expression. So that's sort of like problem one. Given a target, how do you know if it's going to be safe? How do you know if it's going to be effective? Problem two is stepping back more broadly in the field of oncology. The field is developing assets twice as fast as it's discovering new targets to build them against. We've got about 5 times more targets today than we did, assets per target today, than we did about a decade ago. And this comes from an analysis McKinsey did that they published a year or two ago. And so you can end the situation as a field where if we're building assets faster than we can find new targets to build them against, everybody's competing essentially in the same patient population. And if you're one of those patients that we have a good target for, great, you're getting a lot of work around your disease. But for a lot of patients who don't have diseases that we know how to target, they're basically left behind. So there's this problem in the field that we're running out of biology to target. And then within that, there's this opportunity around cell-targeted therapies, meaning in oncology, it is a therapeutic that goes to particular cells in the body and kills those cells, and in particular, the malignant cells in a patient's cancer that are driving the disease, and just bringing a cytotoxic function to those cells. So, we're bringing a killing function to just the cells you want. And then within that, there's a question, okay, how do you find those targets in a data-driven way that are going to be most precise to just those cells and avoid every other cell in the body you're trying to avoid, which that's where the single cell sequencing component comes into this. But then gives you a chance to build out a pipeline that doesn't just go after the same targets that everybody else is going after, but really moves the needle for those patients that we don't know how to reach yet. So it was really those two things coming together. First, this problem in the field where we're building assets faster than we're finding new targets to build them against, kind of like too many hammers, not enough nails. And then how do we find those nails? And then specifically using single cell sequencing and mapping out cells in the healthy body and cells in the patient's tumor and building a data-driven platform to find those specific targets to then build out a pipeline that kind of, corrects for that trend as a whole. So those are the two big ideas. The initial spark came from that CD19 work, but has since kind of evolved and morphed over time as well.
And it's amazing to see the original papers that really kicked off the same idea that you and the team really felt motivated to really start up cartography. It's exciting to hear. And diving a little bit deeper into the origin story, as we're discussing cartography and the other side, I really again, the computational backend and experimental techniques that you all are doing to profile normal and cancerous cells is really creating a transformative roadmap. I'm excited to dive into your all's platform overall. But as you alluded a bit to the similarities between, the hammers and the nails of the origins itself, I really wanted to take a step back, Kevin, before we dove into Atlas and your further platforms. Could you provide a bit of context for our audience and share more about the challenges of, specifically drug discovery for cancer targets that we're facing today?
Yeah. Well, so one of them is simply the crowding in the space. As I mentioned, a lot of targets have 10, 50 programs in development against them. And if you think about this from a very macro perspective, we are over-indexed on the same, aspects of biology, same elements of risk and opportunity, and under-indexed on a lot of other spaces. And so big picture, irrespective of any one company doing well, I think that that's kind of an unideal situation because, as I mentioned, we're over-indexed in areas and under-indexed in under other areas. Um, More scientifically, though, I think a lot of the targets people are working on for, again, I put this class of cell-targeted therapies. So CAR T cells, T cell engagers, antibody drug conjugates, monoclonal antibodies, radio nucleotide radiopharmaceuticals, anything with an antibody at one end that brings a cytotoxic function to particular cells. You need those targets to be super precise. They have to be expressed only where you want and nowhere else. And we don't actually have a lot of those types of targets that are known in the field. So a lot of the known targets are on cancer, which is good. That's what you want. But they're also on other cells you're trying to avoid, which is bad. There's pretty notable examples of targets in hematological malignancies, solid tumor indications, where clinical trials have had unexpected and unfortunate off-tumor but on-target toxicities because those targets' expression is not as specific as you would need to have an effective therapy. And so there's a specific challenge of both the toxicity profile of those targets, meaning when you're dosing a patient, you're hitting cells you're trying to avoid. And because you're hitting those cells you're trying to avoid, you're also limiting the efficacy and the potency that you could have because you can only dose as potently as you're able to tolerate in that patient. So that, I'd say specifically is a challenge for a lot of these known targets, is that they're on malignant cells, but they're also on healthy cells that you would want to avoid clinically. And then it limits your ability to actually build a drug that's potent and effective on the tumor because you hit those toxicities.
Right, of course. And I think really the underlying sense within the baseline challenges in immunotherapy, certainly there's a need for a platform like cartography, and it's exciting to see the initial waves of what you and the team have developed. And
Yeah.
so now as we've kind of landscaped the ecosystem as it looks like with so many solutions and assets that are in the clinic right now that need a specific solution to put this together. Really, I wanted to save the floor for you, Kevin, just really talk more about cartography solution and really how it addresses these more broader issues in
Yeah.
immunotherapy in general. We discussed a little bit at how y'all arrived at the cartography solution and a bit of the genesis of the story. But as you were applying this to industry, I mean, what was really the solution that you and the team came up with and said, this is the platform we want to build. This is what we want to scale to really solve. Could you
Yeah.
talk more about the cartography platform that it is today?
Yeah. Well, so it's actually, I mean, conceptually, it's very simple. And build a drug that goes where you want in the body. I mean, conceptually simple problem. To kind of zoom out first and just say it again, there's a lot of modalities the field has developed. And in particular, we're thinking about T cell engagers for our first program. So I'll use that as an example. That bring a cytotoxic function, and half the molecules focused on the cytotoxic side, half the molecules focused on the targeting side. So a bispecific T cell engager is a molecule that binds tumors on one end and T cells on the other end. And so it recruits the killing of the T cells to just those tumor cells. So the big question here is how do you make that killing and function specific to just the cells you're trying to target? And the way that we do that is actually also conceptually simple, although technically ends up becoming challenging pretty quickly, which is you need to understand everything that's expressed on every cell you're trying to avoid and every cell you're trying to target. And if you had that information, right, if you wanted to understand everything in the body, or rather build a drug that was specific, you'd want to understand everything in the body, then you could just look at the data. The data would point you in a particular direction. Okay, this is the most specific target. You could then build an antibody against that and build a therapeutic pipeline around that. And so that's the big picture framing. The Atlas platform that we've built out, which is our main platform, focuses on identifying individual proteins, individual targets that are specific to cells of interest. So the data set that we've built out is a huge differentiator. It's a huge moat. We've got multiple petabytes of data, proprietary collaborations that we've established with academic centers, clinical institutions, be able to get both healthy and tumor tissues. This has been a huge kind of logistical lift to get set up, because as you can imagine, some tissues are easier to get than others, and it's not the most, you can't just go to Amazon and get this, right? I mean, it's a complicated thing for a lot of very important reasons. Patient consent, of course, being a big one. So we've had to spend a lot of time working to set up these collaborations with different centers to get access to fresh, viable tissue. It has to be fresh, viable tissue in most cases, because in order to generate high quality data, you need high quality tissue samples, not just kind of older or archival tissues. So we built out this collaboration network that's enabling us to build this proprietary data set. And effectively, the end result of this is that we have, to our knowledge and from our perspective, the most comprehensive database of every cell in the body, its different cell state, what antigens are expressed there. And so we have this baseline of kind of the quote unquote healthy reference or healthy atlas that lets us say, okay, given a target, where is it going to be expressed in the healthy body, not in a tumor state, but in a healthy body, so we can understand the safety and toxicity and potential applicability of a given target. That's the first bit. The second bit is similar in concept, but applying it to tumor samples. We've built out a large kind of tumor sourcing network. Same thing, built out these collaborations to get access to viable tissue on tumor samples, generating single cell omics data from each of those samples. And then a combined across this data set, again, as I mentioned, multiple petabytes of data. You run into this problem then where you have, literally trillions of data points of every single gene, all the different variants of those genes, splice variants, and every single cell that you've measured. And the only way that you can actually contextualize that data is with machine learning and AI and other methods to condense it, to find the similarities and find the patterns in the data. But then you end up finding this target landscape for any given indication. You can imagine target quality versus patient coverage. So how good is a target versus how many patients is it expressed on? And then what's been incredibly exciting to see is we can actually find in many indications 5 to 10 targets that we think are much more specific to the tumor and much more tractable to develop a therapeutic against. And so the end result of this Atlas platform, data set on one end and kind of tissue samples going into that, target analysis from the computational end, and then these high quality, high specificity targets that we can then build therapeutics around with the antibody engineering platform. And so that's big picture, that the solution, I guess, to that problem of how we can build therapeutics that are more precise is you need your own data set. You need a proprietary data set that is purpose-built exactly for that problem. Otherwise, you're relying on the older data sets everybody's analyzed every which way. You're not going to be able to find anything new. And so that's the core of the Atlas platform. Now, there's kind of one extension that I'll touch on very briefly, which is our Summit platform. Our Summit platform looks at not just one target, but 2 targets together and builds logic gates. So only if a cell expresses target A and target B will the antibody then bind and activate against that cell. And the reason that this matters is because with any given single target, there's a trade-off between patient coverage and target quality. Some targets are very specific, but they only target a small percentage of the patient population. Other targets cover a lot of the patient population, but aren't very specific. And there's a set in the middle that is a sweet spot that are very specific and cover enough patients to really make an impact. But we really were asking the question, how would you build a therapy that covers every single patient indication and is highly specific, as the best potential single target you could build? And the way that you find that is by targeting 2 antigens together, building a logic gate So only if a cell expresses A and B do you then activate. You're expanding the search space from tens of thousands of potential targets in their splice variants to tens of millions of potential combinations. And so it builds that broader search space, a little bit more complex, but ultimately I think a very big payoff. So those are the two big platforms: Atlas platform, single targets, built on proprietary data sets, and Summit platform, target pairs, to really move the needle in the long term.
It's incredible. And thank you again for giving the breakdown of Atlas and Summit. It's an amazing platform to really see it broken down, both really solving a lot of the potential bottlenecks that we're starting to see in the industry. And, conceptually from a top-down perspective, it's just great to hear the details from yourself, Kevin. But really, one question I wanted to extend on this is really seeing the current applications that this platform is currently affecting on this other side. And so, I wanted to take a moment to discuss some of your more recent announcements, specifically with Gilead Sciences, the partnerships that you and the teams have to develop therapies around the most precise tumor targets in adenocarcinoma and triple negative breast cancer. Really, first of all, congrats to yourself and the team. It's an amazing milestone. It's exciting to see you all pairing with such a great organization. But really, could you first maybe discuss more about this partnership, the applications of Atlas and Summit, and just potentially the future partnerships yourself and the team are excited to pursue as you continue to try to disrupt the current works in precision medicine and immunotherapy that's
Yeah. Yeah, well, thank you. I mean, we're thrilled to be working with Gilead and thrilled to be announcing this first collaboration. There's always been this goal, and I guess I'd say promise of the platform, which is that it could do a lot and a lot of indications to find specific targets. And I think as we looked at the data, it became very clear that this hypothesis was bearing out and that there was more than we could ever do ourselves. As a small company, there's only so many things that we can pursue in our pipeline. And there's a lot of other companies with a lot of amazing resources that we could potentially work with. And so it was exciting for us to get this first deal with Gilead announced. We're working together in two indications that have huge unmet need: triple negative breast cancer and lung adenocarcinoma. Adenocarcinoma is the most frequent form of lung cancer within the non-small cell lung cancer subset. Triple negative breast cancer, a subset of breast cancer with not a lot of options, unfortunately, and really high unmet need. And so within this collaboration, cartography is going to be identifying and validating both single targets and target pairs, finding those targets and target pairs that are most selective to those malignant cells and those indications. We're using both our Atlas and Summit platforms to look for those single and target pairs. And then Gilead's able to opt in on multiple targets and then undertake the further work with R&D, commercialization around those programs for those targets. As part of this, we're getting 20 million upfront. We're also eligible for a set of near-term preclinical milestones and downstream milestones and royalties. And so we're very happy. I think it's an agreement that works well for both groups. For us, it lets us expand the impact that we could otherwise have. For Gilead, I think they're able to really make sure that the programs they're building are against the best antigens in those indications. And so I think it's an exciting thing for us to now expand the reach, not just beyond cartography's pipeline, but also now working together with Gilead here.
Again, incredibly exciting and excited to see the future partnerships, organizations that you and the team are excited to pursue on going forward. Really as a halfway point, but as an extension to the amazing milestones you and the team have reached. Always like talking halfway through these episodes about some of the major challenges and accomplishments of each company. And really, Kevin, you've led this really exciting frontier in immuno-oncology, pioneering new discovery methods for antigen targets to, in 2022, really coming out of stealth, launching with 57 million in total funding, and again, this recent collaboration with Gilead. From yourself, just taking a step back, Kevin, what have been some of the greatest challenges you've faced at Cartography?
Well, I would say one of the first things is simply just getting the team together. That's probably, I'd say, generic to any small company, but you're starting with a couple people and an idea, and you've got to figure out a way to get other people to join in and build a team and culture and environment that helps move the science forward. And so that's been something that I think has been both a challenge and also something that I'm just really proud of how we've done at Cartography. We have an amazing team, incredibly smart, motivated people coming from academic backgrounds, industry backgrounds, different perspectives, and bring it all together in the company. And so that's been something that I'm very proud of that we've done. On the scientific end, I think the thing that is the opportunity and the challenge of cartography is new targets. And specifically, when you find a new target, what do you do with it? There's broadly the field likes to think about kind of biology risk and technical risk. Technical risk meaning taking a known target and building a new tweak to it, kind of a better hammer against the same nail. New biology meaning new nails, thinking about to decide what hammer to use against it. And because we were starting out with finding new targets, and that's really the kind of core of the initial platform, there was also this question, okay, so what are we going to do with those targets? How are we going to think about building a pipeline around those? Are we just going to partner them? We're going to build it ourselves? What modality would we pick? We spent a lot of time over the past couple of years really thinking about the strategy for that set of questions. We very much believe that in order to really bring forward the promise of the platform, we have to have our own pipeline. We have to kind of put our money where our mouth is, for lack of a better term, and build those programs forward. And also, kind of to the point about Gilead, we know that there's more than we can do ourselves, and so we've always wanted to work with other companies. And so that was kind of the first big decision: is we're going to work ourselves on our own programs and look for opportunities to work with others. And then modality becomes a big question. You have a target, you think it's specific. What modality do you pick? Do you pick, and I'll just make it simple in this case, a cell therapy, a CAR T, a T-cell engager, and an antibody drug conjugate? So which of those would you want to pick? And that's an interesting question. We spent a lot of time on it internally with the scientific advisory board. We initially were thinking about cell therapies just because that's how we got into this. As we thought about the path to building out a company, though, there's simply more validation in solid tumors with antibody-based modalities, T-cell engagers and ADCs, and also the path to building that drug is just more straightforward in those two modalities. Cell therapies, I think, have a huge amount of promise, but the sort of off-the-shelf approaches still need time to really get to the place where anybody can work with them. And so we decided to focus on antibody-based therapies. And then there's some nuance based on targets between T-cell engagers and ADCs, really based on the biology of what's going to work best. But that was more of a strategic question as a company that we had to think through. Okay, so again, we have a platform, we can find targets. What modality are you going to build against for each of those targets? And how do you balance the kind of impact of doing the things with a lot of different people, with just yourself? But that's like one of the thorniest questions that we've had to think through as a company.
And it's such an interesting question, and for lack of that matter, certainly not alone in the amount of companies that go through the strategic decisions around. But it's always a difficult one that I love almost highlighting in a way if we have a moment.
Yeah.
Discuss. I'd love to go deeper, Kevin, and talk about it. From a strategic perspective, as you all discussed, as you're talking about these challenges, do you have advice for future founders, other folks around thinking of these longer term strategic questions, but really more in particular, there's been a series that we've always had with BIOS is, the question around pivoting versus persisting. As you all pivoted away and really focused on antibodies and doubled down, do you have similar advice for founders or other folks that are thinking through similar strategic questions?
Yeah. Well, I think the thing that I would say, somebody who's starting this today, you have a new platform, you have a new approach, you've got a new conceptual way that you can build a drug or whatever it may be. And the question then becomes, what therapeutic program do you build and what indication do you build it against? And I think you got to think about it from two perspectives. The first is from your platform and your technology out, what is most tractable for you? So to use us as an example, like if we find a new target, you can actually move pretty quickly with a cell therapy because a lot of the modality platform is transferable across different programs, meaning you can take a new binder, drop it into a new CAR, drop it into a new cell therapy process. And it's not 100% plug and play, but it's more so than it is for, say, an antibody drug conjugate. So that might tip you toward a cell therapy. Um, but then you got to think about it from the indication kind of back and from the clinician's perspective back. And what would most move the needle for somebody working on that disease, irrespective of what your platform can do for it, just what moves the needle for them? And that's where I think you start to see antibodies really emerge because it's something that's off the shelf. It can be applied across many, many different patients. You don't need to manufacture it for each one. They have certainly shown that they've worked in solid tumors. Another T-cell engagers got approved in solid tumors. And so from that perspective, you actually probably tip toward antibody-based modalities as opposed to cell therapies. So I guess I said that because I think a lot of people, when they start out with a new idea, are coming from that place of, okay, we're the expert here, and go one step forward to say, okay, this is what's going to work best with us. But you also have to think about it from the perspective of kind of the long game, which is if I build this thing, how does it fit into the clinical paradigm? And does it make sense and move the needle there? And if the answer is no, even if it's the best possible thing and a great proof of your platform, if it doesn't move the needle or fit into that paradigm, you're going to have trouble down the road. And so that's, I think, the advice that I would give is think about it both in the perspective of what can you do best that nobody else can, and also what does the field need, irrespective of you, and then find the overlap between those two things. Because maybe it's actually the second best thing that you could do that 100% fits into the clinical paradigm, that is a thing you should do, as opposed to the thing that fits best, but only fits into the clinical paradigm, 10% in hypothetical. So I think you have to think about both of those two sides. And for us, modality was an example of that. Where antibodies really just were the thing that fit the best.
It's incredibly sound advice, Kevin. I really appreciate the tangent here, going through your own mental framework, but also discussing the thematics of how other folks are really should be approaching, actual assets, targets, and focus of the company. It's amazing.
Yeah.
So really to continue on this note, as we're still in a bit of this halfway moment, we've discussed some of the amazing milestones and accomplishments your team has reached over the past years with cartography. But really, I always like just taking a second moment, whether it's the personal side, whether it's been the other announcements or milestones you'd like to touch on with cartography. Flipping the question from challenges now to accomplishments, what do you see as the major accomplishments from your perspective with cartography?
Yeah. Well, the first I'd say is the team. I mean, I mentioned it before, but that to me is the thing that makes me excited every day to come into the office. I mean, a lot of things do, don't get me wrong, but the team aspect of science in industry is something that just you don't get in academia. Everybody working together on the same side to move the needle forward. Doesn't matter if I do it or you do it. Like that aspect of the people and the culture is something that I'm incredibly proud of what we've built at Cartography. I think we've got an amazing group of people, an amazing culture, people coming in who have academic perspectives, who have industry perspectives, who have different modalities and different areas of specialization. And I think it's an amazing group that we've been able to pull together. So that's something that I'm incredibly proud of at Cartography, is just the team that we've built. The other thing is that first drug program that we've got in our pipeline. CB21, it's a T-cell engager. It targets an incredibly precise antigen of colorectal cancer, and one that we think is really going to move the needle and be one of those next kind of big targets in the field. The past few years have been about building the platform, validating the target, building a drug against the target, proving that it works preclinically, now thinking about the path toward the clinic. But the program is wholly built internally. The platform pointed to it. We built the drug. We chose it ourselves. And so, I think that's the other thing that just is a big accomplishment for the company. We've gone from start to kind of halfway, I guess, to now getting to the point about we're thinking about the path to clinic in a pretty short amount of time. And so that's something that I think really speaks to, again, that team and the group of people we have around the table.
Exactly. It's always amazing the team shout out again with the lead program that's coming through. Excited again to follow on the cartography story and continue on. And really, as we continue discussing this, we've talked about cartography, your all's approach to date. I'd love to just continue speaking about this out into the future, beyond the clinic, and your future assets. From your perspective, Kevin, what's coming next for cartography?
Yeah. Well, first thing with the pipeline, I think, is getting that program into the clinic. That's top of mind for us, is getting that first CB21 T cell engager moving forward toward phase one. Beyond that, there's always the platform, of course, as a big focus, expanding that and pushing things into the pipeline from it. We have a set of targets we're really excited about. There's some early data that's incredibly compelling, single targets and target pairs. And so we put a lot more to be building, both on the platform innovation side and in the pipeline. I think there'll be more conversations to be having about potential partnerships, how we think about other companies and how we could work together to really move the needle, in a 1+1=3 setting. So a lot to be excited about on our end, and a lot that we're kind of busy pushing forward.
Oh, certainly. And another strategic question that I thought of while you were discussing the future plans, right? With really any platform company, there's almost always that debate between vertical integration and or a horizontal platform expansion. But as y'all are thinking about the future strategy of cartography, how are y'all thinking about these opportunities in direction of a company as a platform, and
Yeah.
what's kind of the future strategy that you all think with
Yeah.
cartography?
Well, it's a really good question. Kind of coming back to that first challenge, I guess, as a company, of if we're finding new targets, what do we do with them, and how far do we take them? And we thought about, what does it look like if we take one asset all the way on kind of one extreme versus we take 20 programs only to the target stage. And maybe no surprise, but the answer that we think is best is somewhere in the middle. We do want to build up multiple programs in the pipeline, and we are doing that. We also want to make sure that we're moving these programs in a way that we can actually advance them, which is to say, if we built 20 programs and tried to move them all at the same speed, nothing would get far enough to really make an impact, at least in the near term. And so we've been very conscious to make sure that within our pipeline, we have something in the lead, and also we have other things behind that are close enough to where you can actually see what's it going to look like, in the next 6 months, in the next 12 months, as those programs advance. And then more broadly, partnering, I think, is really where we get to, make an impact in that more kind of industry sense. Other indications, the Gilead deal, for instance, you can imagine other non-oncology therapeutic areas still in that frame of specificity. So we're certainly thinking about other places that we can apply our platform, but making sure that within the pipeline that we are building a pipeline, it does have programs that are moving forward. It's got a good balance. It's not too much. It's not too few. Things are spaced out appropriately. And that's been a big focus to make sure that, we have that good strategy as a company.
It's incredibly exciting, and thank you again from your perspective, Kevin. One future question, as you were discussing the team you've curated and put together, always like having kind of questions, especially folks that have translated a company through academia. Given academic research has this dual purpose of advancing our own scientific understanding and providing really a training ground for the next
Yeah.
generation of researchers like yourself, Kevin. How does cartography think about talent and really think about connecting with academia as a company now in industry?
Yeah. Well, I'd say there's a few people that we've been really proud of who have gone on to academia after being at cartography. So we certainly, if people want to come in, for instance, out of undergrad and work and kind of advance in their career in industry, great. And people that want to come in, work a couple of years, and go back and get graduate training in medical school or graduate school, we're also thrilled to get to support that. And so we've had a few people now that have kind of come in, done some work, gone back to academia. We actually had one person who interned, who went back, who came back full time, and now back to academia. So we've had a few people go back and forth. And I do like to think that we can be a training ground for people that are interested to grow in their career, either in academia or in industry, but want to get that industry training. We also do think about publishing. That's something that takes a little bit longer. There's a little bit more kind of complexity as a company to think through, but we do anticipate doing as well as we grow out the company.
Of course, and love seeing the split that y'all have been able to make over the past few years while you've been growing cartography. That's great. And really, as we're discussing the future of cartography and the future of the platform itself, from your perspective, Kevin, I mean, really just going out there before the end of this episode, are there any additional enabling antibody technologies you're excited for, excited about for the space more broadly, excited to integrate or bring into cartography in particular? Any calls to startups or other innovators?
Yeah. Well, I think a really interesting thing is around, I guess I'd say two things that are broadly into the theme of kind of pairs of targets and synergy and multiple ways of engagement. The field has done a lot of work moving from just individual targeting monoclonal antibodies to bispecific antibodies, kind of first and second and multiple generations of T cell engagers. We're now seeing that the most recent advances of bispecific T cell engagers can work in solid tumors, and also that you can make them work sometimes even better by modulating them in terms of how they signal to T cells. And so interesting combinations with molecules that have two stimulatory arms toward T cells, or building two molecules that go in together that stimulate different axes, building logic gates against tumors. I think that this area of kind of more complex antibody engineering is something that the field is really going to see a lot of promise of. Um, that's not to say that what we have today can't work. It certainly can, and we've seen that work in the clinic. The new technology is a little more complicated, as you might expect. But I think there's some really compelling data coming around that as well. So I'm excited for that in the coming years.
Certainly. And it's exciting to see how cartography is planning to continue on that side of antibody engineering to continue innovating and pushing the entire ecosystem forward. So once again, Kevin, it's been amazing to have you. I just wanted to ask a few rapid fire questions and close things off for this episode. From your perspective, Kevin, what would you consider to be the greatest challenges advancing precision medicine in kind of the current field right now?
Yeah. I think the biggest trade-off is patient coverage and potential quality of a given target and how you think about de-risking that target and the biology around it. Those are kind of a few things wrapped together, but you could imagine that if every single patient were the same, this problem would be much easier. Not every patient is the same though. And so given a target that is, in a hypothetical, 60% good but hits 40% of patients, or 40% good and hits 60% of patients, or 80% good and 20%, right? Like, how do you make that decision? And I think that that is the biggest challenge because ideally, if it was really easy to build a drug, you'd find the best targets. Doesn't matter how many patients they hit, and just build them all. But that is hard to do for a lot of kind of obvious reasons around the money it requires to build them all, the regulatory work, the preclinical models, the clinical tests. Um, but how you figure that out, I think that's something that is a big challenge in this space more broadly.
Issues that are currently hitting the space right now, and certainly a lot of issues to be solved in the space, but certainly, as you mentioned, incredibly exciting to see it. One question we usually like to end with this, Kevin, as well, is just really projecting out to the future, getting
Yeah.
beyond some of these maybe initial barriers that might be solved, might not be, and really just to pick your brain here, Kevin. Just had a question from your perspective. Imagine it's 2050. From your perspective, what do you see the antibody engineering space to look like? And from yourself, within the oncology space and immunotherapy, how do you see that space evolving by
Yeah.
2050?
Well, my hope is that going back to something I said at the beginning, rather than having twice as many assets per target developing or building assets twice as fast as we're building our new targets, that sort of it comes back to more of an equilibrium. And we have more targets and more drugs against those targets in the biopharma pipeline that have been approved. And so my hope, is in 2050, a patient can come in, they go to a doctor, they get a test, and there's 50 options, there's 100 options, there's a variety of different targeted therapeutic options based on the antigenic profile of that particular patient and how it fits in broadly. Um, and you can just go and say, okay, this is the best thing for you. And it's kind of that's what we see as that middle ground, I guess, in precision medicine. It's not like you need to be N of one. It is true that if you're willing to go smaller in the patient population, you can often find better drugs. And then we build up more of them over time. So that's my goal for 2050 is that we have more things off the shelf, more biology that we've targeted and drugged, and we can just pull that thing out to the best patient or the best option for that particular patient.
Incredibly insightful, and hopefully looking forward to that future as well myself.
Yeah.
For your side, Kevin, imagine still being 2050. Where do you see cartography?
Yeah. Well, hopefully having multiple drugs that have been approved. I mean, I think that's what got me into this. That's what motivates everybody. We've got a pipeline that looks incredibly promising. And now I think it's up to us to really prove it out. And so hopefully by 2050, we'll have multiple things that have gone through, really be pushing again that lens of specificity and everything that can be done there in oncology. So um, that's a goal, multiple approved drugs. I mean, that'd be a big, it's a big goal, but one that we're certainly motivated to work towards.
100%. And we're looking forward to it, Kevin. While we're wrapping up here, any other closing thoughts, shameless plugs you'd like to share with the audience?
Well, I would say, maybe just to think about kind of how I got into this. Um, for any of the grad students or postdocs people in academia thinking about industry or starting companies, like go explore it. That would be my closing thought. Internet a company, consult for a company, think about starting a company or joining a company. But I think I felt when I was in academia, you were sort of in this not box, but you're in this path toward doing a postdoc and getting a assistant professor position, and moving forward. And nothing wrong with that, but you don't get to see, or at least I didn't get to see what it would be like on the other side. And now I've realized for me, I enjoy it so much more. So that's my closing thought: is for people in academia, try to find a way, if you're interested, to explore something in industry. And if you're curious about starting a company, I'm happy to talk, and a lot of other people are happy to talk as well about what that process is like. Um, but just explore around. That'd be my wish that I had done more of if I were to go back 5, 10 years.
Of course, and love the closing thoughts there, Kevin. It's amazing to see the team that you've put together, and excited to also see the shout out again to future folks that are either starting companies or joining out in industry. Some of the potential of that. So, Kevin,
Yeah.
thank you once again for joining us. It was absolutely amazing to have you. Grateful for your time, and hope to have you on again soon. Thanks again, man.
Yeah, thank you.